Yeah, thanks very much to Trevor and Martin for inviting me. I've really enjoyed this conference. I've enjoyed the talks so far, and I've enjoyed all the discussions with the students, and I'm looking forward to further discussions. So I gave the title of Neuromodulation and Cortical Function when we were planning the meeting, but I realized I should kind of mention that I'm really going to emphasize what I call the love of my life in the introduction, acetylcholine. I really am obsessed with acetylcholine, and most of what I'll talk about is relevant to acetylcholine. But I'll mention that the cellular level effects of acetylcholine that I'll talk about often apply to the other modulators, such as norepinephrine, dopamine, serotonin. And then also I'll talk somewhat more about computational modeling than I might normally. I always talk about the modeling, but I'll talk a little bit more about the techniques, partly because Trevor asked me to focus on that. Oh, and one thing I just wanted to mention is that we just got a new building here. Oops. Just got a new building at Boston University, so moved into that a month ago. I'm not quite sure what's going on with the connection here. So anyway, I just wanted to advertise that I'm the director of the Center for Systems Neuroscience there, and we have a lot of good facilities there. I'm not sure what's going on. Can you tell what's going on with the projector? All right, so these are the 4 topics I'm going to talk about. I'm going to talk about initially kind of a framework about thinking of acetylcholine in terms of the circuit level effects, enhancing dynamics for encoding of new input and attention to new input. And then the lower levels of acetylcholine I'll talk about actually towards the end of the talk in terms of providing dynamics for consolidation during sleep. I'll also talk about modulation of persistent spiking and working memory. And I'll also talk about cholinergic modulation relative to grid cell recording and time cell recording in behaving rats. I think I've been very ambitious, even with 3 hours, about what to fit in. I've tried to cut down the slides as much as possible, but we'll see how much we get through this. So this is really my main take-home message right here in the first slide. All right, looking at cellular level effects in slice preparations and in vivo, it seems as if acetylcholine is changing the dynamics of cortical circuits to enhance the influence of the afferent input to the circuit while reducing the influence of excitatory feedback. So the thalamic input to cortical structures and feed-forward connections into the hippocampus and other structures are enhanced. The influence of this input is enhanced by the presence of acetylcholine. And then the excitatory feedback is clearly suppressed. And in contrast then with lower levels of acetylcholine, you'd have a somewhat weaker influence of the input, though it's still present, and a stronger influence of the excitatory feedback. So this is really my main take-home message about acetylcholine, and I think it does apply particularly to norepinephrine as well. So here's a slide of the sort that you've seen multiple times, the basic idea of the modulatory system. Martin talked about this, the modulatory, the neurons in the basal forebrain, the medial septum provides cholinergic input to the hippocampus and entorhinal cortex, and then other structures like the horizontal limb of the diagonal band and the vertical limb of the diagonal band, and the nucleus basalis of minor to project other cortical regions. And as Martin pointed out, Zaborski has shown that there's often some selectivity of the connectivity from these different cholinergic nuclei. And in addition, there's the pedunculopontine and laterodorsal nucleus in the brainstem, providing innervation of the basal forebrain and the thalamus. I want to mention acetylcholine. If you talk to a physician, they'll mostly think about acetylcholine at the neuromuscular junction or as the transmitter at the preganglionic segment of both components of the autonomic nervous system, and also the transmitter at the postganglionic segment of the parasympathetic nervous system. There's 2 main receptor subtypes, the nicotinic receptor subtypes. Martin mentioned those. In particular, Martin emphasized the alpha-4 beta-2. As you probably all know, this is an ionotropic receptor, so it has a channel in the same structure that has the acetylcholine receptor, and it increases the permeability for cations, so sodium and potassium, as well as calcium in some cases. I'll actually focus primarily on muscarinic receptors in my talk, and this fits well with the framework that Trevor gave you earlier about the muscarinic receptors being similar to other neuromodulatory agents like serotonin and dopamine and having entirely affecting the physiology via G protein linked receptors. So here's an example of the M1 muscarinic receptors. There's actually 5 different types that have been described, M1 through M5. But these have a somewhat slower time course than, for instance, glutamate or nicotinic receptors. So it's on the order of hundreds of milliseconds to tens of seconds. And it's because the M1 receptor and other muscarinic receptors are activating these G protein mechanisms, and in particular, subsets of these M1, M3, and M5 activate phospholipase C, which causes enzymatic transformation of phosphatidylinositol into inositol triphosphate and diacylglycerol, and this activates various intracellular processes. For example, I'll talk quite a bit about the influence of muscarinic receptors on potassium currents. All right, these G protein coupled processes will shut down a lot of different types of potassium currents. This differs from the serotonin 5-HT1A receptor, which actually activates potassium currents. All right, so that's a general framework. And I'll also talk about these potentially broader spatial effects. Martin briefly touched on the idea that there might be volume transmission for acetylcholine effects. So this relates to what Martin was talking about. He actually showed, later in his talk, he showed microdialysis studies from his own lab. Here's an example from another laboratory showing that microdialysis techniques, it's very important to pay attention to the time courses that are plotted. Here is a plot where the microdialysis samples are being plotted every 10 minutes. All right, so a very slow time course. Martin showed one that was every 8 minutes. So that was really, until Martin brought in the amperometry techniques, that was really the state of the art for tracking acetylcholine levels. All right, and you can see that it results in, you see relatively slow changes in concentration of acetylcholine when the animal has salient novel perceptual stimuli in its environment. You get these increases that can last for many minutes, actually, in the microdialysis studies. And another thing I'll talk about, because I'll talk about dynamics during waking and sleep, there's other microdialysis studies that took even longer periods of sampling and have shown quite consistently that acetylcholine levels are high in the cortex during active waking. They go down during quiet waking. So this is when an animal like a rat or a cat is exploring the environment actively. This is when the animal's sitting quietly, grooming, or sometimes eating. And then if it goes into slow wave sleep, you have a dramatic decrease in the levels of acetylcholine. And there's a lot of data supporting that dramatic change. And in fact, there's a lot of data indicating through various techniques that change in acetylcholine levels causes very big increases in excitability of feedback pathways within cortical structures. And I'll talk about that a little more later. Yeah, that's an interesting question. Yeah, so most people have been focusing on correlating it with the different behavioral states of sleep rather than the day-to-day circadian rhythms. I believe there's some data indicating that it will change over the circadian cycle independent of the sleep. Oops, sorry, I'm pointing at you with my laser pointer.
When investigating sleep, do people see that the concentration decreases prior to sleep onset or after?
Yeah, that's been a question for quite some time. So there's efforts. Basically, it looks like it seems to come down. I mean, the animal will initially go into earlier stages, stage 1, stage 2, and it's coming down. But I believe that it actually comes down, coinciding with the onset of the slow wave sleep dynamics. And then particularly with REM sleep, I won't talk a lot about REM sleep, but REM sleep actually involves a big increase back up. That's why REM sleep is sometimes referred to as paradoxical sleep, because the dynamics look like waking. You actually have much higher levels of acetylcholine as well, and people have looked at that as well and seen that there's some evidence that the acetylcholine system is activating, to kind of trigger the onset of REM sleep. All right, so I just wanted to kind of be clear about this difference in timescales relative to what Martin talked about. So this is Martin's data from a review that we wrote together, but this is Martin's data where he is recording with amperometry, and you can see the timescale is seconds here rather than minutes, all right? So this a major advance in the field that he brought in these amperometry techniques and sees these second scale changes in acetylcholine. So this is going up and down over a period of 3 to 4 seconds, as opposed to the microdialysis studies where it's showing a change over 5-minute intervals and many intervals, still going really up more like a 20-minute time scale. All right, so this was, Martin talked about this Parikh et al. Paper. I also want to remind you that in his other work, for instance, he did cholinergic, selective lesions of the cholinergic innervation and saw impairments in the sustained attention task, and he did optogenetic activation of acetylcholine to enhance performance.
So sorry, does this concentration correlate to the level of, say, waking and walking state?
Yeah, so Martin actually has not seen as much of a tonic, or slow changes in acetylcholine levels. So I don't know if you want to comment on that.
We cannot measure slow changes for a number of reasons with this technology. So this doesn't translate into what Mike has discussed so far. We cannot integrate these two things, really. Right? And as I showed yesterday, yes, one rides on top of the other, but
yeah.
Is this basic activity related to cue or reward delivery?
Well, as Martin mentioned, he's arguing that it's really related to the cue in this task, all right? Because it comes up before the reward delivery, so he's really arguing that it's the cue that's triggering this acetylcholine pulse. Well, the expectation, and as he mentioned, the incongruent characteristic, so in fact, it's actually not so much waiting for the reward, because if you get the same cue, consecutively, then you don't get this pulse. So in fact, it's more of the not expecting the reward that causes that pulse of acetylcholine to appear. All right, so that was kind of an overview so far of the time course of the acetylcholine effects, but talking about Martin's work on attention, I really want to emphasize that I've kind of been thinking of these effects of acetylcholine in the cortex in terms of how they could contribute to the modulation of attention. And I'm going to talk a little bit more about Trevor's data on this, and then I'll also talk about encoding from memory. All right, so Trevor Robbins is very famous for the development of this five-choice serial reaction time task, which is one of the major attention tasks used particularly in the animal literature. It shows the animal sitting in a box, and it's attending to these 5 positions, and it needs to note when an individual stimulus light goes on and make the appropriate response for that. And over a very large number of studies, not only his group, but other groups as well, these are all ones from Trevor Robbins' group, but a number of different groups have shown involvement in a number of different neuromodulatory systems, but I want to emphasize that they see impairments with basal forebrain lesions that are then reduced with drugs like physostigmine and nicotine, suggesting that the impairment is a cholinergic impairment. They've also done microdialysis in this study and seen that the acetylcholine levels seem to be contributing to the sustained attention in this task. So I think there's very robust data for a role of acetylcholine in attention in the human subjects as well. If you give human subjects nicotine, it enhances their performance in sustained attention tasks, and scopolamine will, in general, cause reductions in attentional performance. But then the other aspect, and I'm not going to contrast memory versus attention, even though some people like to do that, but I'm just going to say that there also are these very interesting effects on memory. Going back really for, actually a century, I would say physicians knew that when they gave scopolamine in preparation for surgery to shut down the parasympathetic nervous system, to block the postsynaptic effects, they would often see amnesia in the patients. And even before that, even in Roman times, it was known that there were certain plants that would actually cause amnesia, and these are plants that contain scopolamine or atropine. Right, so this is an effect that's been known for centuries, and right at the end I'll actually talk a little bit more about the fact that at high doses it'll cause delirium and a complete cognitive breakdown, and I'll show you an interesting video of that, so that's something you have to look forward to. But at lower doses, without causing this cognitive breakdown, scopolamine will impair encoding. So here's a test showing selective effects on encoding. So they first test encoding of a list of words in control conditions, and then pre-injection conditions, and then they give a scopolamine injection or a saline injection. And when they test retrieval of this list that was learned beforehand, there's no effect. If anything, there's actually a slight increase in the number of words that are retrieved after scopolamine has been given. All right, so there's really no impairment of retrieval. But then if they're given another series of words, and this was a large number of words, it was 145 words in each of these lists, here they would retrieve a good performance is about 45 words, all right, in both saline and scopolamine. Here they encode. The saline subjects have a slight, bit of fatigue effect, so maybe they go down to 40 words. the scopolamine effect is really one of the most dramatic behavioral effects out there. They go down to about 5 words on average, all right? So it's really a very dramatic impairment of the encoding of what I would refer to as an episodic memory task, the ability to remember these words in a particular time and place, all right?
Does it matter if the word list is presented as a written list or already there?
It's been done both ways, both auditory and written, but it's not just, they don't view the whole list. They actually get it usually for 2-second intervals, either visually or auditory. And it's been shown that it doesn't impair semantic memory, so it doesn't impair your memory for general facts about the world. It doesn't impair procedural memory. It doesn't impair your learning of motor skills, at least not in the experiments that I'm citing here.
Sorry, does it impair any kind of sort of language function in any way?
I think there's subtle effects on maybe on really long interval language function. That's been shown, for instance, with patient HM, that if there's a very complex sentence structure, you can get influences on that.
I was just thinking perhaps that might in itself affect encoding.
Yeah, and that could be, but I think really these effects are at a dose and a level where it's really quite a simple task normally. I don't think it's a language problem, though certainly at higher levels you will see effects.
And can we say that this result is because of precision memory is more needed to attention, and acetylcholine is more responsible to attentional processor?
I don't quite understand that question.
For precision memory, you need to more attention.
But procedural and semantic memory are not impaired here. So, but the way these tasks were done, they weren't really attentionally demanding. I can see if you set up a particular paradigm, you could have kind of an attentionally demanding task. But the way these were set up, they weren't particularly attentionally demanding. So I don't think that's a connection. And I just want to mention, I'll talk quite a bit about the hippocampus and entorhinal cortex because those structures, as you probably all know, are implicated in episodic memory. So you probably all know the famous case of patient HM who had bilateral removal of the entorhinal cortex and hippocampus that caused a severe impairment of encoding of new episodic memories, but not the retrieval of older and consolidated memories. And this is defined specifically, episodic memory is memory for events at a specific place and time, so not semantic memory. And then in terms of the entorhinal cortex and hippocampus, a lot of the innervation comes from the medial septum, and I'll talk a little bit later about how it's involved, could be involved in grid cell and time cell coding. All right, so just to kind of hammer in my basic message, here we are with the slide again. So now I'm going to talk about what motivated this particular hypothesis. It was partly the behavioral data, but it was also based on very consistent effects in slice preparations, not only from my own and in vivo preparations, all right? So that's part of the reason I'm so in love with acetylcholine and particularly muscarinic receptors, is the data is tremendously reliable, reproduced in literally hundreds of papers by different physiological groups, all right? So there's really, as far as I'm concerned, at least in terms of slice data, there's no ambiguity about these effects. It's a little bit more of a question of the time course of these effects within intact animals, but they're quite robust effects. So these are slice preparations. So here's a case where we're preparing slices where we rapidly dissect a brain out of an animal that's been decapitated. We have to dissect it out in about 4 minutes, put it into the slice chamber where we're keeping it alive with artificial cerebrospinal fluid that is oxygenated and has glucose. So we're essentially keeping these cells alive in the slice preparation for many hours, up to 6 hours or so. And then we can go and we can record. In this case, we're showing, actually, this is a neuron that's been filled with biocytin and fixed later. So that's not the way it looks in the slice, but it's just so you can visualize the individual neuron. And we're recording intracellularly. And this shows that we can, if we give a bit of current injection, we can push it up to cross threshold so it would actually generate action potentials. So these lines are intermittent action potentials. And there's actually interesting subthreshold dynamics as well. So this is what is being done to measure a lot of the effects that I'll be talking about in this section of the talk. And so here's an overview, and there's a lot of different effects, but I like to put them in this framework of enhancing the input and suppressing the feedback. All right, so the enhancement of input, there's actually a direct enhancement of thalamocortical transmission that I'll mention in the slide, but there's also an enhancement of the spiking response of the input through the depolarization of cortical pyramidal cells and a reduction in spike frequency adaptation. And then later I'll talk about this suppression excitatory feedback, which is due to presynaptic inhibition of glutamatergic transmission. And finally, I'll talk about mechanisms that could enhance encoding, such as increased long-term potentiation and increased persistent spiking. But first, I'm talking about these effects. And these, no matter what modulator you're working on, these are relevant to you because norepinephrine does this same reduction of spike frequency adaptation. Serotonin causes the same reduction of spike frequency adaptation for certain subtypes of receptors. And dopamine, there's evidence that that also modulates the spike frequency adaptation. So these are really important to you no matter what modulator you're working on. So here's the basic effect. Acetylcholine suppresses one thing, so in control conditions in the slice, the cells tend to be quite hyperpolarized. But if you put on acetylcholine or a cholinergic agonist, such as carbachol, it will cause a long-term depolarization of the cells. It'll come on over several seconds and last for an extended period of time. All right, so these receptors don't seem to habituate like the nicotinic receptors. And this is a very robust effect that's been shown both in vivo and in vitro, literally in hundreds of papers. This is due to a block of a resting potassium current. So the potassium current has a Nernst potential at -90. When it's open, it's pushing the membrane potential down towards -90. When you block it, it's going to depolarize. Here's an effect, another effect of potassium currents is that during spiking activity, there's activation of calcium channels, voltage-sensitive calcium channels, and you get an influx of calcium during each of these spikes that activates calcium-sensitive potassium channels, often referred to as AHP currents, and this will actually hyperpolarize the cell so that it stops spiking, and that's referred to as spike frequency adaptation or spike frequency accommodation. All right, so the cells won't, when you're giving a tonic current injection here, but the cell fires initially and then slows down and stops firing. But if you put acetylcholine or carbachol into the slice chamber, the same current injection now gives a higher frequency of firing that is persistent through the whole period of the current injection. And this is because you have a decrease in this calcium-dependent potassium current. And again, like I said, this is a very well-replicated effect. And as I also mentioned, norepinephrine does the same thing. Madison and Nickel showed that as well, and serotonin also does this for certain receptor subtypes. And dopamine is a little bit less clear, but you do see effects. All right, so these are very general effects, and if you're thinking about modulatory effects in the cortex, it's very useful to know about these effects. So because Trevor asked me to talk about modeling, I'm going to give just a bit of an overview about how we can model it. The bottom line is that we can very effectively model these effects. So this just shows, just skipping ahead, that we can develop biophysical simulations that can replicate the spikes and replicate the slowing of the spiking, the spike frequency adaptation, and then we can reduce the calcium-dependent potassium current and get more sustained spiking activity. And this is using one particular technique in computational neuroscience, which is using the Hodgkin-Huxley formalism developed in 1952 for modeling the dynamics of voltage-sensitive channels. And this was initially developed by Hodgkin and Huxley to model the dynamics of action potentials. So there's very complex equations. This is essentially an equivalent circuit model of the membrane potential with the essentially the Nernst potentials are like batteries driving the potential, and then their influence on the potential is proportional to the conductance or resistance for particular ions, sodium, potassium, chloride. Uh-oh. That's not a... And then you can combine these conductance together along with the resting conductance. And then the changes in these conductances, the G's are representing conductance. The changes are modeled by Hodgkin and Huxley as being dependent on the current voltage of the membrane. So it's a very complex set of differential equations. They can't be solved analytically, but they're solved with numerical simulations with very short time steps. And you can get quite good replications of individual action potentials. And this is used not only for the spiking channels, but also for other channels like the M current. That can use it. You can have calcium in the equation and have it activate the calcium-dependent potassium current. you'll have multiple of these circuits, so one for the soma, and then you can either replicate all of the dendritic compartments or simplify it down into maybe 3 compartments for the apical dendrite and a couple of other compartments for basal dendrites. And this just shows examples of simulated neurons, showing that you can simulate the suprathreshold activity, the spiking activity, as well as the subthreshold activity. Here's another example of a simulation of a neuron showing it's spiking with both the suprathreshold activity and, in this case, subthreshold oscillations. So that's a general technique that can be used for analyzing how neuromodulatory influences on channels can influence the dynamics of the circuit. And it's, as a very simple outcome in looking at the suppressant of spike frequency accommodation, you can see that when acetylcholine is present, you have more spikes being generated in response to a particular input. And in a simple associative memory model using these spiking biophysically detailed models, the larger number of spikes causes more conjunction of pre- and postsynaptic activity, and therefore more strengthening of synaptic connections. So in a sense, enhancing encoding just by enhancing the spiking response of the neurons. And you can also think of this in terms of attention. If there's a more robust spiking response to the attentional input, then you're going to have a better likelihood of detecting that input. So it's really quite a simple framework, but I think it's useful because essentially it's connecting things from the molecular cellular level all the way up to the behavior.
Sorry, if the computation at that particular area of the brain is not dense coding, but rather sparse coding, would this be like opposite of like for a good information processing task?
You mean would it impair information processing, or?
Like this would be true for C3 or C1 regions, but maybe in the pairs you have sparse coding. So in this case, like amplifying the response will not be exactly efficient for the encoding of the information.
Yeah, no, I see what you're getting at, and certainly a lot of circuits are, sparse coding circuits. They have feature detectors that are, quite sensitive to its individual features. And so this particular example, and a lot of the examples I'll show are these very simple associative memories, but at least for the influence of acetylcholine on the circuit properties, my sense is that it does translate pretty well to sparse coding and primary sensory cortices, and there's even evidence from the Teal laboratory in Scotland, Herreras et al., where they showed that it influences the response of visually responsive neurons and seems to give more end stopping, for example. So, quite robust feature selective responses are modulated in a similar way, where it seems to be enhancing the response to the input and reducing the internal spread that would, maybe cause more of a spread beyond the end of a particular visual stimulus.
So I'm maybe not getting this. Why would it, so even if this is happening in a network that implements sparse coding, the fact that you enhance a particular salient or important input, then why should that be a problem? I mean,
why should it be a problem for sparse coding? I'm not sure, but I don't know where you're directing your question, at her question or at me. Yeah. I mean, I could see a reason why, just enhancing the overall activity to the response to the input could be a problem. If you're only, if you're just pushing the whole curve up evenly, you certainly could reduce the ability to discriminate particular stimuli. But I think that's,
doing it in a precise way.
yes, it's certainly, that's my sense of it. It should help if it's precise. And I think the suppression of the excitatory feedback helps, in a sense, to balance out the enhancement of the response. And I'd actually like to see more people working on this. There are people in V1 working on cholinergic modulation, but I think we need more people working on that. All right, so again, back to how this fits into the framework. Now, I've shown physiologically how this acetylcholine effect enhances the influence of afferent input. Now I'm going to move to talking about the reduction of the excitatory feedback. All right. And on an intuitive level, you've even I think you've experienced this. At the beginning of a talk, you have high levels of acetylcholine, you remember what the person's saying, and then as the talk goes on, you probably get these intruding thoughts coming in. You think, oh, well, what I'm going to have for dinner, what I'm going to do at the break. You start to get maybe more, excitatory feedback activating other thoughts. That's a very simplistic way of thinking about it, but I think it's relevant. And so in a sense, when you're in the most tuned in, you have the most high levels of acetylcholine. In a sense, you have a block of some of these internal processing dynamics.
So how does this correlate also
Yeah, well, so the idea is that during active waking, the animal is attending more to the environmental stimuli and encoding more stimuli. So behaviorally, it's important for it to be attending, whereas when it's sitting quietly and grooming, it actually doesn't really need to bring in the information in the same way. So that's the basic idea. So now I'm going to talk about this reduction of excitatory feedback in terms of presynaptic inhibition to glutamatergic transmission. And now, before my talk, how many people had heard of this, that acetylcholine can inhibit glutamatergic transmission? Raise your hand if you've actually heard about that. Yeah, so this is what I kind of expect. It's really not, within the general neuroscience community, it's not what people think about. If they think about presynaptic inhibition, they'll usually think about autoreceptor effects, which Martin mentioned when in his talk he talked about nicotinic autoreceptors reducing acetylcholine release. So acetylcholine reducing acetylcholine release. All right, but this is a case where acetylcholine is modulating glutamate release. And it's really not well known in the community, but again, there's actually at least 100 papers on this topic for various modulators. So this is showing how we can look at this. A simple way to look at it is we take slice preparations of different structures. We've done it for hippocampus, olfactory cortex. It's been done in neocortex. This is just my kind of clearest slide was piriform cortex. And what's nice about piriform cortex and hippocampus is that the afferent fibers bringing input to the structure are actually segregated into the distal layer contacting distal dendrites of the pyramidal cells. So this is input from the olfactory bulb, all right, into the piriform olfactory cortex, and the neurons are contacting distal layers so you can record synaptic potentials induced by stimulating the afferent fibers. Or you can record potentials induced by stimulating the excitatory feedback connections, which terminate in the more proximal region. In piriform cortex, the same dynamics are in region CA3. Right? So we can separately study these pathways by stimulating these different fiber layers and looking either extracellularly at the synaptic potentials or intracellular. So here you can record the intracellular membrane potential changes for both of these separate synaptic inputs. And the simple message is, as I've mentioned, excitatory synaptic glutamatergic feedback is suppressed by acetylcholine. All right, so here's two potentials in control conditions for the afferent input and the excitatory feedback. And with a cholinergic agonist such as carbachol, there's actually not much change in the afferent input in the piriform cortex. In other structures, such as the neocortex, there's actually a nicotinic-mediated enhancement of synaptic potentials that have been shown by a number of groups, including Changeux and Barry Connors and so on. And then during the same manipulation, the excitatory feedback, here's the synaptic potential in control, here's during the cholinergic bath application, and you see a dramatic reduction in the size of the synaptic potential. Wouter?
And how long does this inhibition of excitatory feedback remain if you have a given perturbation?
Yeah, so it doesn't seem to habituate. So initially this was bath application, and so it would turn on with the infusion of the carbachol or acetylcholine, usually with an acetylcholinesterase blocker if you use acetylcholine. And then it'll last as long as the substance is present. And in fact, the biggest difficulty is when you wash out the carbachol or acetylcholine, you often don't have complete washout. So Alfredo Kirkwood has proposed that there's actually a cholinergic long-term depression of the synapses. But it isn't. This is an acute effect. This effect comes on over a matter of seconds and it's been shown that it's a presynaptic reduction of the release of acetylcholine.
Are these useful for
Yeah, so that's an interesting question. Unfortunately, carbachol, if you put carbachol onto cortical preparations in rodents, it'll actually cause epilepsy rather than blocking it. And so it's this relative balance. It's probably that it's increasing the excitability more than it's suppressing the excitatory feedback. So.
I guess it's known what is the receptors that mediate presynaptic effect.
That I've got. So let's see, is that the next one? Yeah. So here it's an M4 muscarinic receptor, and this is something that I looked at before they had the particular selective knockouts. We tried to look at it pharmacologically, and it was difficult. But now there have been selective knockouts showing that this presynaptic inhibition at the M4 receptor, at the excitatory feedback connections, is mediated by acetylcholine activating presynaptic M4 receptors that mediate this presynaptic inhibition. So if you have a knockout of M4, you almost completely lose that presynaptic inhibition.
Would you see a paired pulse stimulus?
And if you do paired pulse stimulation, then what you see is that you have a stronger reduction of the first stimulus than the second one, which again indicates that it's presynaptic, that it looks like there's still calcium rushing into the presynaptic terminal, but then it's causing actually more release for the second pulse. And let's see if I could even show, I guess not. I have a slide showing that in hippocampus, there's actually a selective distribution of M4 receptors at this proximal layer, the stratum radiatum layer, where these connections synapse, as opposed to the afferent input, where there isn't as much M4, but as I mentioned, there's actually evidence that nicotine will enhance synaptic transmission in that layer.
Have you also done any experiments for nicotinic receptors?
Yeah. So people have done nicotinic experiments, and as I mentioned, nicotinic experiments are a little bit less consistent in their effects. One of the most surprising things is that you don't see fast or what you would expect. You'd expect ionotropic effects, and instead, when they see effects, it's due to like long-term ionotropic application of nicotine, at least in terms of modulating synaptic transmission. You can see fast excitatory effects postsynaptically in some preparations. It's much more difficult to see those, partly because of the desensitization. But Changeux has shown, and Danny, and a number of people have shown nicotinic enhancement of particularly thalamocortical input in areas like prefrontal cortex and somatosensory cortex and auditory cortex. So this does seem to be relatively robust nicotinic enhancement of glutamate release at those input connections.
Would you define this effect like an example of a G-protein receptor that is actually pretty fast effect?
Well, let's see. I mean, I'll show you actually data where we did a pulse of acetylcholine. In fact, it might come up soon, but we did a pulse of acetylcholine followed 100 milliseconds later by atropine to block the receptor. And the effect still came on over 2 or 3 seconds. So it still seems to be slow, like a G-protein coupled effect. And then I just wanted to mention this postsynaptic effect is traditionally considered to be primarily due to M1 receptors, at least on the pyramidal cell, so that the reduction in spike frequency accommodation. And this has, I think this has really interesting clinical implications in that I believe that selective M4 agonists could be quite useful for treating of Alzheimer's disease and potentially for schizophrenia, and particularly since those diseases are associated with hyperactivity in the hippocampus in early stages. And so if you can give these M4 agonists, I think it can reduce, and there's some evidence that it can have a positive effect. So this is just summarizing the same thing I already said. So people like Shirey and Gullidge have shown that the presynaptic inhibition is due to activation of presynaptic M4 receptors that directly cause a reduction in glutamate release. And it actually doesn't seem to be blocking calcium influx, it seems to be blocking the machinery for glutamate release. All right, and I just want to emphasize that this is a very consistent, well-replicated effect. So I've been emphasizing the work I did showing the selectivity for feedback versus afferent input, but earlier Hounsgaard, and I wanted to mention Hounsgaard because he's Danish and there's a lot of Danes here, he had a paper on this effect in the Schaefer collaterals and stratum radiatum of region CA1, Valentino and Dingeldine. Then these papers are ones where I showed that it was selective, so that it was affecting stratum radiatum, but not the input from entorhinal cortex and lacunosa moleculare. Vogt and Reger showed it for the recurrent excitatory connections in CA3 and stratum radiatum, but not for stratum lucidum input from dentate gyrus. And that fits, it seems like a different connectivity because the lucidum connections are very proximal, but again, they're providing input to CA3 from dentate gyrus, so that is not suppressed, but the excitatory recurrent connections from CA3 cells to other CA3 cells could be thought of as excitatory feedback. And then it's been shown in a number of neocortical structures, somatosensory cortex, visual cortex, auditory cortex, by a number of different groups. So again, I'd like to really emphasize that though it's not well known within any population, it's well replicated. And this is just showing essentially the same thing laid out in the connections to kind of emphasize that people tend to think of the hippocampus sitting at the top of the hierarchy of cortical connections. So you have feed-forward connections coming in through the neocortex into the hippocampus to CA3, and then you have strong feedback connections there, and then feedback connections back to the neocortex. and the feedback certainly is very strongly suppressed in CA3 and from CA3 to CA1, and then back into the neocortex. This is a study that was done many years ago in vivo, showing that during different levels of cholinergic modulation, during, for instance, slow wave sleep, they see much more dramatic spread of activity through the hippocampal circuit. And then these are these cortical studies. And then just to mention, so this framework I gave you, I focused on acetylcholine, but many of these effects are appearing with other modulators. So suppression of spike frequency accommodation occurs similarly with norepinephrine as well as serotonin. The presynaptic inhibition of transmission occurs with GABA B receptors activated by baclofen, as well as alpha-1 adrenergic receptors that will inhibit glutamate transmission. And then, interestingly, metabotropic glutamate receptors, which have the same G-protein coupling, there are some subtypes that selectively inhibit the afferent input. So that does happen in some cases. Okay, so I'm just going to briefly mention that consistent with the enhancement of encoding, acetylcholine enhances long-term potentiation. This has been shown in a number of studies. I'm just going to very briefly, just for time's sake, just show an example where, in control conditions, you get a certain amount of enhancement of the height of EPSPs, and with a cholinergic agonist, you'll get a larger enhancement of the size of synaptic potentials, even though there's an acute suppression, there's an acute presynaptic inhibition. So when you put on acetylcholine, the synaptic potential is going to get smaller, all right? But then after you wash it out, you're going to have more LTP than if you just did the stimulation without it, all right? All right, and then I'm just showing you that quickly here. Right here, you put on carbachol, you get the acute presynaptic inhibition, but then afterwards you actually see the LTP. All right, and the amount of LTP, even though you're suppressing it during carbachol, the amount of LTP is larger than you would get without the acetylcholine.
The increase in amplitude is still presence of the carbachol?
No, so we've washed it out. Actually, yeah, the line isn't clear here, but the carbachol is only here during this time, and then once it's washed out, you lose the acute presynaptic inhibition, and you see visibly the long-term potentiation. Okay, so now I'm going to talk. I've talked about these.
So the potentiation is of the synapse that involves the afferent influx, or is it also the?
Yeah, the excitatory feedback connections. In the slide I just showed, this was at the excitatory feedback connections associated with afferent input. You can do it different ways, but it actually, the modulation actually seems to be stronger at the excitatory feedback connections. And that's a great lead-in, because so it suggests this kind of paradox. Why would the cholinergic enhancement of LTP be stronger at the connections that see presynaptic inhibition? Or vice versa, if you're enhancing those connections long-term, why are you suppressing them? All right, and here I have a really nice, clear story from modeling. It was entirely the modeling literature that gave me this story, and it wasn't, I didn't just invent this. When I found this selective cholinergic effect, I looked at all the literature, the neural network literature that existed. You've probably all heard of Hopfield nets. That's one of the most famous neural network models. But there were other people like Anderson and Cahonen that had used similar associative memory models. And I was very pleased to find, it took me a little while to realize it, but I was very pleased to find that they were essentially implementing exactly what acetylcholine does in their models without ever giving a physiological justification for it. So I'll show you examples of how this works in the slides, but just to kind of quickly mention Hopfield nets, what he does is he clamps the activity of the network to the input and computes the outer product of that vector of input, all right? So he completely, in his model, he completely ignores the excitatory recurrent connections when he's encoding a new pattern, as if he knew that acetylcholine was causing presynaptic inhibition to those connections, which of course he wasn't thinking about that. But he had to do it for mathematical reasons. He had to clamp the network to encode, and then when he wanted to do retrieval, of course he had to turn on the excitatory recurrent connections to get the attractor dynamics. So he did exactly what acetylcholine does. And it was there in all of these other models for mathematical reasons, not for physiological reasons.
So what acetylcholine does on the main input, not on the feedback side, what it does on the main glutamatergic?
Yeah, it's enhancing it, if anything. It's making it stronger. Yeah, well, yeah, it's enhancing the thalamic input or the input from the olfactory bulb to the piriform cortex. And I'll show you why that's important to have that. All right, so here's an example. Now, I hope this is clear. So what I'm going to do is I'm going to put it in the context of a behavioral task, the standard paired associate memory task, to try to give you a sense of why this is important, why it was done for instance, in the Hopfield net. So the paired associate memory task is a task where subjects learn a series of word pairs, and often they're related word pairs because that makes it easier for the person to learn, but they're not learning the meaning of these words, they're just learning these two words were associated on this day in this room. So it's an episodic task. So that's the context, and they're given, for instance, a word pair leather with holster, all right, and I'm showing how you would model this in a Hopfield net or other associative memory. You have input activating different populations of neurons. So each of these circles, these filled circles, represent active populations of neurons. And then you do Hebbian long-term potentiation. So as you know, or like spike timing dependent plasticity. It's dependent on pre- and postsynaptic activity being conjunctive. And that causes strengthening of these connections between the coactive neurons. So neurons that fire together, wire together. And then during retrieval, in that context, that first word is given, what was associated with the word leather, and the activity spreads across these strengthened connections to activate this population, and then the activity would spread out to your language cortex from the hippocampus, and you'd be able to generate the word holster. And this is pretty clearly, it has to be happening in hippocampus or entorhinal cortex because patient HM can't do this task, all right? So if, it's happening somewhere in the structures that were removed from his brain.
Is there any particular meaning to this particular choice of the network itself? I mean, why is it that you have 2 layers into?
Yeah, in this case, my effort was just to have the simplest example to show the network dynamics. Obviously, the hippocampus is more complex, so here I'm just trying to show the simplest case of input connections, providing the input to the circuit, and then essentially I consider these excitatory feedback connections between the neurons within the population. And it was really just for illustrative purposes. But it's similar, you could think of it as similar to the structure of region CA3 of the hippocampus.
So the ingredients that you need, so the simplest ones are context nodes and input nodes and output nodes?
Yeah, I mean, for this particular model, but I think it's more general than that in the sense that CA3, most people who have modeled episodic memory in the hippocampus do have neurons that are getting some representation of context, neurons that are getting some representation of the stimulus. I wouldn't make a distinction between input and output. I could have had connections going both directions here. So these are all, this is providing input to the circuit, all right, and then we're strengthening the connections. But then once there's this completion effect, then that would go out through region CA1 and back to the entorhinal cortex.
Excuse me. The affinity of the receptors involved in two opposed mechanisms. It's the same or is different?
You mean like the nicotinic versus muscarinic receptors?
Yeah.
Yeah.
These receptors involved in LTP.
Oh, I see. For LTP, yeah, versus presynaptic inhibition. Yeah, that's an interesting question. These are M4 receptors. These seem to be more probably M1 receptors, and so the affinities are actually somewhat different between those receptors. So there could be, slightly differential influences depending on concentration. All right, so this framework works just fine for single word pair, but now I'm going to show the phenomena of proactive interference, which is, imagine you're getting that same word, or not the same word, you're getting the same first word, but a different second word, all right? So you get the word leather associated with the word boot in a different context. Often in these experiments, it's just 10 minutes later, all right? So it's a different temporal context, all right? But you can think for episodic memory, you can meet a friend of yours, here in this room or down in the, in the lunchroom, and those are very different contexts spatially as well. But here we, just to illustrate what happens in, in the Hopfield net, in the Conan networks, whatever they are, when you try to learn a new association, you have the problem that the previous association can be retrieved. So the word leather comes in, and the activity spreads across this strengthened connection to activate the population of neurons representing the word holster, all right? So in a sense, that's interference from retrieval of the previous association. And so now you're trying to store this association, but instead you get this activity. It might inhibit the activity that you want, and you'll get an association between the new context and this earlier word. So the context 2 will get associated with the word holster that wasn't used in that context. So this is an undesired strengthened connection. So now when you give the word leather in the second context, you might get an intrusion of the previously stored word. You might end up saying the word holster instead of the word boot. And that's the basic idea in all of these circuit models. I just used a behavioral task to kind of give you an intuitive sense of it. And now this problem will actually build up pretty systematically as well. And so here's where having the effect of acetylcholine is appropriate, because you now can prevent that interference from retrieval by presynaptic inhibition of the excitatory recurrent connections. So the same connections that need to be modified are being inhibited presynaptically. All right, so this is the same associate, the second associate. You have the word leather coming in, you have the word boot coming in, but now we're applying presynaptic inhibition of glutamatergic transmission only at these internal feedback connections in stratum radiatum, not the afferent input in lacunosum moleculare or lucidum, wherever it's coming from. All right, so you get the input coming in actually stronger. The input comes in and clamps the activity to the pattern that you want, and you don't have any interference from retrieval. And at the same time, acetylcholine, there's still some transmission. It's not completely blocked, all right? So there's still activation of postsynaptic NMDA receptors, and there's enhancement of NMDA currents. You actually, and that's probably the mechanism for enhancement of long-term potentiation. So you're blocking interference, but you're enhancing the encoding for the connections that are being driven by the external input, all right? And that will give you much more solid coding of this new association without interference from the previous association.
Have you looked at postsynaptic NMDA receptors to make sure the acetylcholine are really reduced in time from anyone?
To see if they're sufficiently activated? Or what did you say about the postsynaptic NMDA receptors?
Have you looked at those receptors?
So, yeah, I mean, that's certainly been done extensively for LTP studies. They can block them with, 2-amino-5-phosphonolvaleric acid, so they can block the NMDA receptors and show an absence of LTP. And then people have done studies of that sort to, influence the LTP in vivo preparations. It hasn't been specifically done with cholinergic manipulations. So this is, I've shown a relatively simple example, but we've actually incorporated this into a network simulation of the hippocampus with all the different subregions, really storing the associations in CA3 and then having regulation. In this older model, we actually had feedback regulation of acetylcholine from the medial septum to set the dynamics for encoding of new information. Now, as Martin mentioned, that's maybe too long a pathway really to be regulating, and it could be that there's actually local circuit regulation, as Martin said, of the acetylcholine release locally within the hippocampus to set the dynamics for encoding. And a prediction of this model, and this is kind of to relate to up to the human level, a prediction of this model then is that scopolamine ought to cause an enhancement of proactive interference. And so we tested that. We actually had subjects learn a series of word pairs, what I'm referring to as AB, that includes word pairs like leather holster. Then they're either given scopolamine or saline, and then they're given either overlapping word pairs like leather boot or non-overlapping word pairs that they hadn't seen previously like kitchen spoon. And in control conditions, the subjects in this case did pretty similarly on the overlapping and the non-overlapping word pairs. But then when they encoded under the influence of scopolamine, they actually showed a stronger impairment for the overlapping word pairs than for the non-overlapping word pairs, suggesting that scopolamine was enhancing proactive interference.
What if you had to just learn one context and a pair of words and there were no sequential overlapping or non-overlapping variants? Would you need acetylcholine for that?
Yeah, it's an interesting question. You actually still see an impairment of just baseline performance, so that would be like here where you don't have the overlap at all, and there still is an impairment. So, and that's probably due, to reduction of long-term potentiation, for example, or just a reduction of the amount of postsynaptic spiking activity. So there is an effect independent of the proactive interference. So it's really taking whatever background effect there is, and it's not really a background effect, there's a clear effect on memory, plus you have an enhancement of proactive interference. All right, and then just to mention, I just want to kind of connect this back to the biophysical simulation. So we've done this in network simulations with these compartmental models that I've shown before. This actually looks very similar to the diagram that Trevor showed at the beginning about these kind of network connections which can be modeled. And this just shows the basic idea of this associative completion. So it's the same idea, but instead here we're coding a pattern of activity across a circuit of these biophysical neurons. So these black squares represent neurons that are spiking theta rhythmically in response to rhythmic input. And then we test the memory function by giving an incomplete memory cue. And I think probably most of you have heard of the idea of region CA3 of hippocampus mediating pattern completion. And here you can see this model very effectively does pattern completion. So this missing component of the input is completed by the model. There's a spiking activity due to the spread on recurrent connections. And this is if you do the learning with acetylcholine, but if you do it without acetylcholine, this is actually multiple learning of multiple different patterns. You can see you get much more broadly distributed activity. So you have a buildup of interference. And I would argue, I'm not going to really emphasize that too much today, but I would argue that this could be what happens in Alzheimer's disease, that you have this buildup of interference, and actually there's quite a bit of robust fMRI activity showing early hyperactivity in people with mild cognitive impairment. And in fact, I guess I should have put in the slide, my wife Chantal Stern had a study where working with one of her students who was Colombian, she went to Colombia and worked on a family that had a PS1 mutation. So they knew these subjects with this mutation would develop Alzheimer's disease and the controls wouldn't. And they found hyperactivity in the people with the PS1 mutation in the hippocampus during memory and coding tasks. So I would argue that this sort of thing is happening even in pre-symptomatic people that are going to develop Alzheimer's disease. So it's a different framework. Often people will talk about excitotoxicity, but I think that it's actually could be due to this phenomena, runaway interference. And this just illustrates that. So I've already shown you this basic idea. For two associations, you get one undesired connection, but for a third association that overlaps with the second association, now you actually add two additional undesired connections, right? And acetylcholine can prevent that. But I just want to show how that builds up over time. So if you have acetylcholine present, you just strengthen the connections you need. If you don't have acetylcholine present, not only do the two patterns interfere with each other, but the interference builds up. So you end up, in this case, with 5 times as many synaptic connections strengthened within the circuit. All right? So this is why I would argue that they should try M4 agonists for treatment of Alzheimer's disease to prevent this runaway interference.
So in the presence of acetylcholine, LTD would not happen?
LTD? Actually, acetylcholine enhances both LTP and LTD, all right? So it's been shown, I mean, those are different stimulation protocols, but it'll actually enhance both. And that would, it actually would even help further if you're, here without acetylcholine, you actually get interference and you don't have LTD, all right? Whereas here with acetylcholine, we actually had LTD in this model. You're enhancing LTP when you need it, but then you're also enhancing LTD to get rid of these undesired connections. So acetylcholine is very good for encoding in these models. And of course, it's been shown behaviorally in a number of studies. All right, I'm just to kind of get back to what Trevor wanted in terms of connecting to different models. I've shown you these biophysically detailed models, but I also want to relate it to the more general class like the Hopfield net or the Cohen networks, these what are called continuous firing rate models. And I just want to kind of give it an idea of the different types of models you can work with. They don't all have to be biophysically realistic because the biophysically realistic models take a long time to run on the computer. They're more complex to develop and they're much slower to simulate. So what you can do is do what are called continuous firing rate models, which if you read about the current explosion of interest in neural network models like deep learning, those are all continuous firing rate models. And the basic idea of those models is instead of modeling each of the individual spikes of a neuron, instead they simulate that as a continuous change in firing rate. So here's a classic physiological study of what's called an FI curve, which is the frequency of response for different current injections. So you give a low level of current injection, the neuron is slightly depolarized, generates 2 spikes. Here it generates 10 spikes, then it goes up and up. As you give more and more current injection, you get more and more spiking. And it looks like this with below threshold, you get no spiking, and then the spiking goes up almost linearly, though it eventually saturates. All right, so you get an increase in firing rate with amount of input. And that's what's actually used in a lot of these deep learning networks now. They have what are called threshold linear units, where below the threshold they don't fire at all, and then above the threshold they basically just have a linear increase in firing rate based on the input. And this is just the way that's represented mathematically.
So this model doesn't consider at all the inhibition, I mean the negative?
No, in this case, this is just talking about the input-output function for a single neuron, but I'll talk a little bit later, if I have time, about feedback inhibition and how it can influence the output of the whole circuit. Because it's essential, you can't just, if you just have a bunch of these neurons together with excitatory feedback, the network will instantly explode. All right, so you have to have that feedback inhibition and some kind of attractor dynamic. And in fact, I think that might even be here. Yeah, okay, good lead in. All right, so if you just have excitatory feedback, then it's a little bit hard to see, but if you have only excitatory feedback, so instance, if your excitatory connections have a certain value and you don't have any feedback inhibition, the network will just exponentially explode. All right. But if you have some feedback where the activity of these neurons now in continuous firing rate mode is activating inhibitory neurons, and then these are causing feedback inhibition, so hyperpolarizing synaptic inputs on these excitatory cells, then you can actually balance things so that initially the feedback excitation is driving the network higher, but then all the inhibitory neurons come over threshold and they turn on and they drive it back down, and then they get weak enough that the excitatory takes over and so on. So in this case, I purposely had it oscillate into what's called a fixed point attractor. So if you hear of attractor models, they have different dynamics, but the basic idea is that they're dynamically evolving into a steady state. And that's often plotted here. You can plot the excitatory neuron activity versus an inhibitory neuron activity and see initially it's just the excitatory activity, and then the interneuron comes over a threshold and it starts to influence the activity and brings it back down, and then essentially this oscillation here is spiraling into a fixed point attractor that has a certain value of excitatory activity and inhibitory activity. And even if you take away the input, this can be maintained in a sustained manner because it goes into another stable fixed point attractor. All right, and I'll mention this a little bit later when I talk about working memory, because this is actually the standard model of working memory, using different parameters and different structures, but that's the basic idea of most working memory models, is that excitatory feedback is maintaining the activity during an extended period. So I just wanted to show, I can show exactly the same effect I showed for the more biologically detailed model. Here I have an input pattern. I give an incomplete version of that pattern. This is, during learning with acetylcholine, all right? And then when I've learned it with acetylcholine, then I give an incomplete version, actually, in this case with less acetylcholine present, and I get completion. So here's the input pattern, and then here's evolving over time the completion of the missing elements of that pattern. And if I do it, if I have multiple patterns stored and I try to do the same thing without acetylcholine, I get this buildup of interference. And like I said, that's why Hopfield designed the dynamics the way he did, because these models will do that automatically if you don't block the excitatory feedback during encoding. All right, so that was just an effort to kind of show you the full scope of different types of models. I think we have maybe 10 more minutes till coffee break. I wanted to mention that you might be thinking, well, I've talked about acetylcholine being important for encoding, and that maybe lower levels of acetylcholine are important for retrieval. And so initially, I thought, when I found these effects, I thought, oh, well, it'd be really cool if cholinergic neurons could turn it off and on really quickly. And this was years before Martin had shown with amperometry these fast pulses of acetylcholine, which I think is what really is needed, all right? But it's still an interesting question, because you asked about the time constants of the G-protein coupled effects. And we actually looked at this. This is what I mentioned earlier, that we gave a pulse of acetylcholine, pressure pulse application, followed by a pulse of atropine through a different pulse pipette. And this is showing extracellularly recorded synaptic potentials, and they're at full height. It's a little bit confusing because the extracellular potentials are downward directed, but we're plotting their height relative to the baseline. And here's the pulse at time 0, and you can see it actually gets the presynaptic inhibition building up over 3 or 4 seconds and then lasting for 10 or 20 seconds. All right, and that's, I think the acetylcholine effects I've talked about are still relevant in the sense that you are going to have enhanced encoding when you have these, this tonic suppression of excitatory feedback, but you might think you also need to have some rapid dynamics between encoding and retrieval. And one possibility is that GABA B receptors actually have a faster time course of effects, though it's still, still coming down over 1 second and coming back up again. But the other possibility is that you could be doing encoding and retrieval at different phases of the theta rhythm oscillation. So Trevor mentioned that I would talk about how theta is influenced by acetylcholine. So you have the medial septum providing both cholinergic and GABAergic and glutamatergic input to the hippocampus and the entorhinal cortex. And it's been shown that both the cholinergic and the GABAergic input is important to theta rhythm. The one thing that happens is the GABAergic input is actually rhythmically inhibiting. There's rhythmically active GABAergic neurons that are inhibiting GABAergic interneurons in the hippocampus that are thereby disinhibiting the pyramidal cells rhythmically, and that could contribute to the oscillations. But then you also have cholinergic modulation more slowly depolarizing the pyramidal cells, and that basically allows the theta rhythm to be expressed. So if you block acetylcholine, if you lesion acetylcholine, you have less theta rhythm power. And I'll actually show if you inactivate the medial septum, you lose theta rhythm. And what's interesting about it is that within each cycle of the theta rhythm, if you just look at 2 cycles, this is the field potential, you can take the field potential at each layer across CA1 through pyramidale into stratum radiatum into lacunosa moleculare. So this is showing at one phase of theta, this is 2 cycles of theta, but at one phase of theta, there's very strong synaptic current sinks from entorhinal cortex and outward current at the cell body, and at a different phase there's very strong sinks in stratum radiatum, and this is the phase when you have inward current at the cell body indicating that spiking activity is occurring. So this fits the dynamics I just showed you. There's one phase where there's strong input and no postsynaptic spiking, and a different phase where there's strong input at the excitatory recurrence, and you have postsynaptic spiking that could give you readout of the retrieval. So that's the basic idea of this model. It's essentially the same model on a faster time scale where the encoding would be happening at one phase where the entorhinal input is strong. It's been shown that also there's a phasic difference for the induction of LTP at one phase of theta. And then during retrieval, you'd have weaker entorhinal input, but you'd have these strong synaptic currents in stratum radiatum, and you'd have less LTP, all right? So it suggests that you could have these encoding and retrieval dynamics on a fast time course during theta. And related to that, Laura Colgan in the Moser laboratory showed that fast gamma rhythm oscillations are correlated between the entorhinal cortex and the hippocampus at what I would call the encoding phase of theta, all right? And then at a different phase of theta, slower gamma in CA3 correlates with CA1. And this is just showing that you do have this very systematic, this is actually recordings in entorhinal cortex showing that in entorhinal cortex as well, the fast gamma is at a particular phase of theta. So this relates to what Martin was talking about, the phase-coupled oscillations, the relationship between gamma and theta. And we see, not a huge change, but a significant change in this phase-coupled gamma relative to theta after, in this case, systemic scopolamine injection. So I don't think it's not quite as nice as Martin's data in that sense. But it's consistent with the idea that maybe the gamma is contributing to the entorhinal driving of hippocampal activity during the encoding phase. Are there any questions? All right, so I'm going to move on to talking about the cholinergic modulation of persistent spiking. I guess maybe they haven't set up. I'll go for another 5 minutes just to be on time, and you can kind of have the basic idea of the persistent spiking. So this is relevant. I'm going to couch it primarily in terms of working memory. So long-term episodic memory involves coding information at one point in time. It's assumed synaptically, and then often in the free recall tasks you don't do anything or you have some interfering distractor task for 10 minutes, and then you're tested for your retrieval. All right, so that's what's needed in an episodic memory task. But if you want to hold the information online, that would be working memory, particularly if you were going to do manipulations on it, as Trevor pointed out. So this is another effect that could enhance encoding both for long-term episodic memory, but also for working memory. It's this idea of persistent spiking. So most physiologists, when they think about the response of a neuron to current injection, they'll look at the current injection, and then the neuron is spiking just during that current injection. But then when the current injection ends, the neuron, in many cases in cortical structures, will show this long after hyperpolarization because of the calcium-dependent potassium current. So that's kind of the standard physiologist view of what a cortical neuron should do, but Angel Alonso at McGill University showed this interesting persistent spiking effect, which has been replicated now in a number of different structures, where if you have acetylcholine activation of muscarinic receptors, either with carbachol or with acetylcholine, the neuron, as I've shown you before, will fire more spikes. There's spiking here. They're all kind of clumped together because it's a really wide time base, right? But there's more spikes here than there are here, just as I've shown you before. But what's interesting is when the current injection stops, instead of showing this slow after hyperpolarization, instead the neuron actually has an after depolarization that comes up and generates spiking activity. And in a sense, the balance of this after depolarization and the after hyperpolarization allows the neuron to show sustained firing at a similar frequency for an extended period of time. Here it's being shown for about 45 seconds, but he actually showed that this bistable activity could persist for 30 minutes. So it's a very robust effect in the slice. The one problem is we haven't yet shown this exact effect in vivo. But the currents are there. So I'm going to just show, this is a recording on an oscilloscope. I don't want it to be too loud, but you'll see this is the baseline, and then there's a current injection, and you'll hear the spikes. All right. So there was the current injection. You heard the spikes during the current injection, and now what you hear are each of those pops is the sound of a single spike being fed through an amplifier, into a speaker, and you can hear that it's a relatively steady firing. In fact, it's right around theta frequency in this particular neuron, right? And it can maintain that, there's no further input to it. It's not getting any further current injection. So previously it was not firing, gets the current injection, and now the same level of current injection that was here, now the neuron is in this bistable state. You can see its membrane potential is staying much higher, and it's continued to spike in a relatively rhythmic manner due to this interaction of the depolarizing and hyperpolarizing currents.
I wonder if you looked at the extracellular concentration.
That's an interesting question. It's been done. One question would be, well, is it due to synaptic transmission, circuit dynamics? It's not, because you can get the effect with AMPA blockers and NMDA blockers. So this is an intrinsic mechanism. It's not a network mechanism. So it's not the same as most working memory models where it's excitatory feedback. It is an interesting question. I guess there's the possibility that you're slightly changing the extracellular concentration of potassium, for instance. That's actually an interesting idea, which we haven't really systematically studied. But this is the basic idea, is that you have cholinergic modulation activating a particular channel called the calcium-sensitive non-specific cation current. So this is a depolarizing channel. It's like a nicotine receptor in the sense that it's letting both sodium and potassium through, so it'll be depolarizing. That depolarization will cause spikes, that'll cause calcium influx, that'll further activate it. And that is one component of the mechanism of maintaining that activity. And I think it's exactly time to head for coffee. So why don't we go ahead and do that?
That's great.
Yeah, thank you.